Hello everyone, welcome back to my channel Xgene and ProteinX. In this tutorial video, I'm going to talk about the transfection. Mainly, I will talk the principle as well as all of the steps regarding transfection, and the transient and stable transfection I'll discuss here. Let's begin. So, what is transfection? So, transfection is the process of introducing a purified nucleic acid, for example DNA or RNA, to the eukaryotic cells. So if you introduce any DNA or RNA into the eukaryotic cells, that process is known as the transfection. So to perform the transfection, you have to some important points you have to keep in mind, that the cell viability, you have to check the cell viability. It should be higher viability, around 90% viability. It will be better. And obviously, when you subculture the cells for transfection, that and just before transfection, the cell confluency should be not more than 50%, and cell should be a single cell and uniformly distributed in the dish. So you have to keep in mind that point. So it may your transfection efficiency will be high. So reagents needed for the transfection: antibiotic and serum-free media, PEI, and nucleic acid, DNA or RNA, whatever it is. The role of PEI, so mainly the PEI is the transfection reagent, and DNA and PEI, it form a complex, and this complex, it carries to the nucleus. So basically, the PEI helps DNA to take the DNA to the nucleus by forming the complex, and that complex is known as the polyplex. Let's have a look at the protocol. So the PI that I mentioned earlier, the role of the PI. So the plasmid and PI, this ratio should be 1:3. So this is optimized. We optimized in our lab. So first, in a 1.5 ml sterile tube, you have to take the plasmid. The plasmid should be sterile, obviously. And if I consider the plasmid concentration is 2 microgram per microliter, and this protocol is for 10 centimeter dish. So then 6 microliter plasmid you have to take here. So in total 12 microgram. And then add 494 microliter media without antibiotic and serum free. So this is antibiotic and serum free media I have to take. So in total it will be 500 microliter, right? And in another new tube, you have to take the sterile PEI. And if I consider the PEI concentration 2 microgram per microliter, then 18 microliter of the PEI. So in total 36 microgram. So here 12 microgram plasmid and 36 microgram PEI. So 1:3. And then I have to add 482 microliter media, the same condition without antibiotic and serum. So this is the preparation test. In one new tube contain plasmid with media, and another new tube contain PEI with media. And then the plasmid and media that you prepared in the earlier step, and that one you need to transfer the entire all of the volume, like 500 microliter, into the PEI media, that tube. So you have to transfer it from here to PEI tube. So in total it will be 1 ml. So it will be 1 ml. Then leave it for 15 minutes at room temperature to form a DNA PEI polyplex, to form the complex that is known as a polyplex. So it's in total volume it should be 1 ml. And then you have to take your dish from the incubator and discard the media completely. Make sure that there is no serum or no media left, because serum, it influences the transfection efficiency. And then wash twice with sterile PBS or serum free media to wash out complete serum from the dish, and then add the polyplex, that DNA PI that we left it for 15 minutes. So after 15 minutes, you need to add this polyplex completely, like 1 ml, to transfer to this dish that contain your cell, and then add 3 to 4 ml pre-warmed. This is you have to keep in mind that it must be pre-warmed, that pre-warmed in 37-degree temperature, and then add 3 to 4 ml pre-warmed media without antibiotic and serum, and then just shake it very gently 2 or 3 times to mix it properly, and then you have to place it in the incubator for overnight, like 12 hours, and next day after 12 hours you have to replace the media with complete media, so that contain no serum. So then you have to next day you have to replace the media with serum media, and then after 24 hours you have to check it under microscope, and then if your plasmid contain EGFP, then you will see the green fluorescence under fluorescence microscope. So this is how green fluorescence, and this green fluorescence it indicating that the efficiency of your transfection. So higher this green fluorescence means your transfection efficiency very high. So next I will discuss about what is transient and what is stable transfection. So transient transfection, actually it is a transfected material enter cells but does not get integrated into the cellular genome. So that's why in the cell, when the cell divide in the next generation, this gene, the transfected genes, it won't transfer to the next daughter cells. So it won't pass these transfected genes into the daughter cells. So as you see here, so this is the transfected genes. The genes is here, it's not integrated into the genome. So when cell divide in the next generation, it won't pass into the next daughter cells. In other hand, the stable transfection, the plasmid DNA successfully integrate into the cellular genome, and it will pass successfully into the next generation of the daughter cells. So as you see here, the transfected genes is integrated into the genome. This is why when the cell divide in the next generation, it will pass into the daughter cells. This is the main difference. So this is actually very easy to develop. It around 1 week it takes to develop, but this is very lengthy process. It takes several months and there are a lot of steps. So it does not need any selectable marker, but it need selectable marker, and it expresses actually lower yield of protein compared to the stable transfection, but this one it expresses higher yield of protein compared to the stable transfection. And for this one, for protein purification, same transfected cells you cannot use more than one time, but here the same stable transfected cell you can use multiple times, so you can save it, you can store it in the nitrogen, no problem. So next I will talk about the steps of stable transfection development. So for the stable transfection, you have to have the selectable marker like puro resistant or geosyn resistance or whatever it is. So it must contain that kind of selectable marker. So here it is puro R, right? So what you have to do? So first, after transfection and after 12 hours, you have to replace media with complete media, that is the same as And then after 48 hours, replace media with complete media with selectable marker. So from here, it would start the stable transfection step. So here you have to add selectable marker, and from this step, whenever you do all of the steps, you have to use selectable marker, for example puromycin or geosine. And next step, after 48 hours of the subculture, you have to seed from here 100 microliter only into new 10 centimeter dish. So rest of the cells you can save it, or you can just keep doing just for one dish. But 100 microliter from out of there, you have to transfer into new 10 centimeter dish, and you have to, by pipetting, you have to single cell you have to make it, and the cells should be uniformly distributed, and then allow to grow these single cells to form clones. And then peaks, when you check the under microscope and when you see that clones are big enough, then you have to pick some good clones into 24 to 40 old dish. So around, it's up to you that how many good clones you can pick. So one clones in each oil, in one well, and then in the 24 or 48 wells dish, you have to allow this clone to grow. It takes several days. So after that, you have to select some oils in the 24 or 48 oils, and then from that oil you have to choose some good oils containing bright clones, and then from there you have to transfer clones into 10 centimeter dish, like one oil to 1 10 centimeter dish. So like this way you can do 5 or 6 or 7 dishes. And then this cycle, this whole entire cycle, you have to carry out like 4 to 5 cycles. The same process you have to do 4 to 5 cycles. So then when you observe that after several generations, still all of the cells are expressing green fluorescence, that means that your stable gene expression already done, that already developed. So you see in this figure that all of these clones it expressing the green fluorescence. So it means that all of the cell contain these transfected genes. Alright, so this is how it can perform. So next is the factors influence transfection efficiency. There are some factors that may influence crucially on transfection efficiency. So first is that I mentioned that PEI. So in place of PEI also very good for transfection, but if you want to get higher transfection efficiency more than that compared to the PEI, you can use the lipofectamine. And you have to make sure that the cells that you use, that not more than 30 passages you subcultured, and also do not allow the cells to remain confluent for more than 24 hours. And also the presence of serum, it influences DNA PI polyplex formation. So you have to make sure that there no serum is present there. And also the presence of antibiotics, it leads to lower transfection efficiency. So you have to make sure that that your dish contains no antibiotic. And the quality and size of the DNA, if the DNA or plasmid is very high and big size, then it may lower the transfection efficiency. And also the quality of the DNA, you have to check plasmid, purification, everything you have to go thoroughly. And also the confluency and viability of the cells, that's important factors, because higher confluency it decreases the transfection efficiency. And also the viability, it should be 90%. And also the cell condition, if like cell grow fast, then obviously the cell condition is good, so you have to check those factors. So this is the another important point, that if you use 293 cells for transfection and to get higher protein expression, then you have to use freestyle 293 expression medium. that is higher expression of the protein. Normally in industry or for higher protein expression, people use this kind of media. So this is all about the transfection. I hope this video will be helpful. So if you like this video, kindly hit the like button, share it, and please subscribe my channel. Thanks.